So in yesterday's class, we studied basics about protein. Okay. I'm briefing what I did in the last class. First of all, we see proteins. These are made up of amino acids, and the basic structure was this. And when the proteins are arranged in this form by the help of condensation reaction, means amino acid 1 plus amino acid 2 plus amino acid 3, it leads to the formation of peptide bond between them. This was peptide bond. See, this peptide bond is planar and it shows partial double bond character. Is it fine? These are the two resonating structures which results into the partial double bond character between the carbon and nitrogen bond. Is it fine? And when amino acids are like joined together in this form, in the left side lies the N terminal, means this amino group. Okay, Gunjan. I said proteins are polymers of amino acids. The basic monomer or the repeating unit here is the amino acid. And by the condensation reaction, two amino acids combine to form a peptide bond. And this peptide bond, that is CONH, is planar in nature. It shows partial double bond character. And these are two resonating structures. The double bond here comes to this C and bond, which shows partial double bond characteristics. Again, I said when amino acids are joined linearly, on the left hand side lies the N terminal. And if there is N terminal or N terminus, which group lies left hand side? That is amino group. And on the right hand side, means this side, lies the carboxy terminal. It means the carboxyl group lies on the right hand side. Now, there was a nomenclature that was introduced by Emil Fischer. For absolute configuration. And this was for the carbon at asymmetric carbon atom only. Means it must consist of asymmetric carbon atom or the chiral compound. Am I audible now? Is my voice still breaking?
Yes.
Okay. So I said Emil Fischer introduced the nomenclature for absolute configuration, and this configuration was relative around the asymmetric carbon atom only, means the carbon must consist of different atoms. And this type of carbon is known as chiral carbon. Is it fine? I'm giving a statement here. Is this true or false? This was asked in GATE this year only. Okay, Ikra said false, Richa said true, true except for glycine. But this statement was general. So we know glycine is achiral. So this statement here is false. No, no. This is the general statement. We can't leave exceptions here. We know glycine is achiral. So this statement is false. So, according to Emil Fischer, he gave the absolute configuration for glyceraldehyde based on the position of OH. If it is on the left hand side, then it is L-glyceraldehyde. And if the same OH is on the right hand side, it is D. The same goes for amino acids. This CHO group is replaced by COOH, and instead of OH here we have amine group. So the relative position of amine group is considered in place of OH. This is L amino acid and this is D amino acid. Is it fine? So what these D and L isomers are? These are non-superimposable mirror images of each other. So these are enantiomers. These are enantiomers. D and L forms are enantiomers. Again, the same question was asked in GAT. The statement was D and L enantiomers are epimer, anomer, enantiomer, and achiral. These were the statements and the options. So the right option was enantiomers. And here I wish to give a statement that in living system we have only L amino acids. हमारी human body में there are only L amino acids present, and generally L amino acids in a protein will react with only L amino acids, and the reason for this presence is still not clear, but there is a theory that L amino acids are more soluble. This is the first theory. L amino acids are more soluble than D amino acids. Hence, in due course of evolution, only L amino acids are chosen. And second thing I said, uh, L amino acids will react with only L amino acids, due to which if in a racemic mixture that consists of both forms, D and L forms, there will be a complexity. There will be complexity among amino acids. Neha said, so we do not consider glycine. We do consider glycine. That is why that statement was false. And now Ashu said, repeat it, sir. What I have to repeat? I said in living system there is presence of L amino acids only. The first theory was L amino acids are generally more soluble than D amino acids. So in due course of evolution, this solubility concept for L amino acids were chosen. Second thing I said कि L form will only react with L form. Okay, it will not react with D form. So if they're in a racemic mixture, uh, there is presence of both D and L amino acids, it will further lead to complexity. And to avoid complexity, only one type of form is present, and that is L form. There is exception to this also. I'm telling in the next slide. I said L amino acids are present only, but there is an exception. In some bacteria, D amino acids are present and they also code for proteins. So particularly, there are antibiotics or drugs that are designed that consist of D amino acids. So this particular antibiotic will be specific to only bacteria because D form will only react with D form. And the example of such antibiotic is ibuprofen. This is very deep thing. This won't be asked, but I'm generally telling. But in the like pharmacology, there is a particular chapter that involves the formation of drugs related to their configuration. That is related to their absolute configuration. So there is another thing. These capital D and L, are these similar to small d and L? Not every bacteria consists of like D amino acids. There are some group of bacteria that consist of D amino acids. So we are targeting that D amino acids by these type of antibiotics. And now Anju is asking, sir, what if bacteria contains opposite forms of amino acids? There are various antibiotics that are designed that specifically attacks bacteria. For example, the cell wall of bacteria, that is quite different for our. Do animals consist of cell walls? No. तो जो भी वो एंटीबायोटिक होगी वो सिर्फ बैक्टीरिया पे अटैक करेगी. ये सब हम देखेंगे, ये माइक्रोबायोलॉजी की थिंग है, ये हम देखेंगे डिफरेंट क्लासिफिकेशन ऑफ एंटीबायोटिक्स. तो आई आस्क्ड हियर, क्या ये दोनों सिमिलर चीजें हैं? कैपिटल डी, कैपिटल एल एंड स्मॉल डी स्मॉल एल. Somebody said not getting the point. So the point here is in the second theory, I said the configurations that are similar, they will react with each other. Means L amino acids will form bonds with other L amino acids only. Similarly, D amino acids will react with D amino acids, and this configuration to this D form of amino acids will relate with D form of carbohydrates. So this antibiotic is designed to attack this type of amino acids. Am I audible now? I was not speaking earlier. Am I audible now?
Yes, sir.
Okay. So I will discuss this thing later. Let's not waste our time on this thing as it was like additional thing I was telling. I said capital D and capital L, these represents the absolute configuration, but these small letters, these represents, small d represents the dextrorotatory thing, means the bending of light towards the right hand side and the bending of light towards the left hand side. So these two things are different, and it is not necessary that capital D form of amino acids are only dextrorotatory and L are labiorotatory. These are not necessary things. They can be different or they can be similar. Is it clear now? So let us discuss today the amino acids hydrophobic, the amino acids that consist of hydrophobic side chains. The category today we will be discussing is somebody wrote a very big message. Right hand side. Today these are the amino acids that consist of hydrophobic side chains. They do not have polarity or charge in their side chains, and the mnemonic for them was GAV limb, means glycine, alanine, valine, leucine, isoleucine, methionine, and proline. So let us start with glycine. Three-letter code was GLY and single letter code is GO. So glycine being the smallest amino acid and the simplest one, it is non-chiral and so no optical activity. Is this point clear? Smallest, simplest, non-chiral or achiral simply, no optical activity. It acts as inhibitory neurotransmitter and it provides central So these are the points for glycine. Glycine is smallest, simplest, non-chiral, and no optical activity because here this hydrogen and this hydrogen are similar atoms. The, it leads to it's achiral in nature. Then if it is achiral, no optical activity. The second point is it acts as inhibitory neurotransmitter. Maybe Tanya ma'am will discuss this thing. Another thing, the third point is it provides central C2N subunit in purines. So somebody said something. O stands for optical activity. Okay, in nucleic acids, there lies purines and pyrimidines, means the nitrogen जो सेंट्रल C2N सब यूनिट्स होती है प्रीन सिंथेसिस की वो ग्लाइसिन से आती है तो प्यूरिन्स हमारे पास दो होते हैं एडेनिन एंड वेलिन Am I audible now? I have written that C2N subunit that is involved in purines, it comes from glycine. Next is alanine. Last point, okay. In last point, I said glycine amino acid provides C2N subunit that is the basic backbone in the purines, and there are two purines, adenine and guanine. Next we have is alanine. In alanine, we have CH3 here. C2N means दो कार्बन और एक नाइट्रोजन. So the function of alanine is it is involved in glucose alanine cycle. This we will discuss in detail. Means it carries ammonia from muscles to liver. When we will discuss the protein metabolism, we will see the glucose alanine cycle there. Next amino acid we have is valine. It is branched chain amino acid. It is important as if it is Am I audible? Am I audible? Okay. So there is a disease that is sickle cell anemia, which is caused by point mutation in GUG. This is a genetic codon. It is converted into GAG. GUG codes for glutamic acid, and it is converted into GAG that codes for valine. And which disease is this? Sickle cell anemia. So just with change of single amino acid from glutamic acid to valine, the structure of hemoglobin disrupts. As here, the nature of amino acid is changed. Valine is hydrophobic in nature. Even this question was asked in this year GATE only. Next we have is leucine. Okay, I'll repeat it. See, I said GUG is converted into GAG. Glutamic acid is converted into valine. See, if glutamic acid is converted into somehow aspartic acid, the structure will remain somehow the similar because both of them are glutamic and aspartic. Both of these are aspartic amino acidic amino acids, but valine here is hydrophobic in nature. Due to its change, the structure of protein is disrupted and it is causing a major disease that was sickle cell anemia. Again, here it is branched chain amino acid. GAG. For valine, it's GAG. And leucine is promoter of muscle development. And prevents muscle damage. Next we have is isoleucine. See, अब तक जो भी हम अमाइनो एसिड्स देख रहे थे उनमें जो 3 लेटर कोड था उनके फर्स्ट 3 लेटर्स ही थे, फर्स्ट 3 अल्फाबेट्स ही थे, बट आइसोल्यूसिन का होता है IN। यहाँ चेंज है, बट आइडेंटिफाइबल है। इससे हमें पता चल रहा है कि कौन सा अमाइनो एसिड। It provides hydrophobicity to that particular amino acid. ब्रांच्ड चेन जो भी है वो उसको हाइड्रोफोबिक नेचर प्रोवाइड कर रहा है, वो ही उसकी सिग्निफिकेंस है. So even this is branched chain amino acid. These were all the branched chain amino acids that were leucine, isoleucine, and valine. So there is a disease that is caused by the accumulation of branched chain amino acids, that is maple syrup urine disease. Maple. This is a disease that is particularly a metabolic disorder, and it is caused by the deficiency of branched chain alpha keto acid dehydrogenase complex. Branched chain alpha keto dehydrogenase complex. This is the enzyme, and due to its deficiency, accumulation of branched chain amino acids take place. And कौन से branched chain amino acids? Leucine, isoleucine, and valine. And in this case, death may occur due to swelling of brain and seizures. It is also called branched chain ketoacidosis. It is autosomal recessive in nature. This thing we will discuss in genetics. I said there is a disease that is caused by the accumulation of branched chain amino acids, that is maple syrup urine disease. And in this disease, there is deficiency of branched chain alpha keto dehydrogenase complex. And this disease follows the pattern of autosomal recessive disorder. Is it clear? Is it clear? I'm just briefing what is autosomal recessive. The genetic disorders can be related or present on the autosomes as well as the sex chromosomes. So in case of autosomal recessive disorders, that disorder is passed from like parents to offspring, not in a dominant fashion, but it in recessive fashion, it can skip generations too. And in detail, we will discuss this thing in genetics unit. Next amino acid that we have is methionine. Methionine is a special one. Why? Because it consists of sulfur, यहाँ से भी एक क्वेश्चन आया था लास्ट इसी ईयर कि जो डाइसल्फाइड बॉन्ड्स होते हैं वो किस किस के बीच में फॉर्म होते हैं। तो सल्फर कंटेनिंग दो अमाइनो एसिड है, फर्स्ट इज मिथ्योनिन एंड अनदर वन इज सिस्टीन। तो ऑप्शंस थे सिस्टीन सिस्टीन, सिस्टीन मिथ्योनिन, मिथ्योनिन मिथ्योनिन and methionine cysteine. These were the options and the correct answer is this. Methionine is not involved in the formation of disulfide bonds. Only cysteine forms disulfide bonds. उसके पास sulfur present है methionine के पास, yes, but वो disulfide bonds form नहीं करता. Is it clear? So methionine का क्या रोल है? It acts as initiator codon. And it is coded by AUG. मीन्स जब भी जो सेंट्रल डोग्मा होता है हमारा डीएनए से आरएनए एंड आरएनए से प्रोटीन फॉर्मेशन, जब यहाँ प्रोटीन फॉर्मेशन होगी तो इनिशिएटर कोडोन विल बी एयूजी एंड इनिशिएटिंग प्रोटीन विल और इनिशिएटिंग अमाइनो एसिड विल बी मिथ्योनिन. Is it fine? Another thing is improper conversion of methionine can lead to atherosclerosis. And methionine is needed to make creatine. Next amino acid that we have is proline. Again, this is a special one. Why? Methionine is needed to make creatine, means it is one of the substrate in the creatine formation. Yes, Anju, I will make the structure of cysteine in like when we discuss cysteine amino acid, I will make the structure. Two cysteine amino acids together, they form cysteine. I will discuss that in next class. This is proline. It is the only amino acid that consists secondary amino group. That consists of secondary amino group. This statement is asked many times, and we call secondary amino group as imino group. And proline, it is the only amino acid that does not form blue purple color with reaction with ninhydrin, it forms yellow color. This will see after, like when we see the protein detection methods, that is there a particular method using ninhydrin. So all amino acids reaction with ninhydrin forms blue purple color, but it forms yellow color. Proline forms yellow color. So only amino acid that do not form blue purple color with ninhydrin. And this proline, it is mostly found in beta terms. This we will discuss in secondary structure of proteins. Is it clear? आज वाले सारे क्लियर हैं? Yellow. It forms yellow. Optical activities like bending of light either in the right side or the left side, dextro or labio. And this is shown by only the chiral compounds. Ninhydrin, nin. And atherosclerosis is just the buildup of cholesterol and fats, and these are built up in the artery walls, and the lumen of artery is somehow decreased. See, this is artery, and in case of atherosclerosis, the accumulation of fat and cholesterol occurs, and this lumen, this area that is called as lumen, it is decreased. And how methionine is involved, that I'll discuss when we will discuss the cholesterol and hypercholesterolemia. In hypercholesterolemia, there is a specific topic of atherosclerosis. I will discuss this thing there. थोड़ा बड़ा टॉपिक है. जस्ट अभी वर्ड डिस्कस किया है तो मैं डिजीज बता रहा हूँ. आर्थेरोस आर्थेरोस्क्लेरोसिस में होता है क्या है, जो आर्टरी की वॉल्स होती हैं उन पे फैट्स और कोलेस्ट्रॉल की डिपोजिशन हो जाती है, जिसकी वजह से आर्टरी का ल्यूमन कम हो जाता है. अगर ल्यूमन कम हो रहा है तो बीपी हाई होगा और हार्ट अटैक के चांसेस बढ़ जाएंगे. ये पर्टिकुलर टॉपिक है हमारा जब हम लिपिड की कैटाबॉलिज्म और एनाबॉलिज्म मींस लिपिड मेटाबॉलिज्म देखेंगे. उधर है हाइपर कोलेस्ट्रोलीमिया, उसमें है ये पूरा टॉपिक. कोई और डाउट्स हैं? हेलो, यस सर. Yes, optical activity shown by chiral compounds only. Okay, कुछ और भी होंगे. So this is all for today. In next class, we will discuss the only classification left, that is hydrophilic side chains, and the hydrophilic can be polar uncharged or ionizable side chains. We will see the positively and negatively charged amino acids. Fine, so that's all for today. Thank you everyone.